Assalamu alaikum. Uh, Achaji, today we will discuss active transport and then bulk transport. Just give me a minute. Let me get ready. Okay. So let's see in active transport. Just give me a minute. Okay. So this is an active transport. Active transport can be described as, active transport can be described as transport of materials into or out of a cell against the concentration gradient. For example, if this is an animal cell and you have sodium ions outside and sodium ions inside. For example, it is high concentration of sodium ions on outside. Maybe we can represent with some number, or we can simply write high concentration on outside and there is low concentration on inside. So this would establish a concentration gradient like this: high concentration on outside and low concentration on inside. Okay. So in nerve cells and some cells, sodium ions are moved out of the cells by an active transport, which involves carrier proteins known as sodium pumps or sodium potassium pumps. So this movement of sodium ions against the concentration gradient from inside out of a cell with the use of an ATP is known as an active transport. Okay. Substances like glucose, amino acids are absorbed in the intestines by an active transport. Similarly, mineral ions like nitrates are absorbed by the root hairs by an active transport. And in the nerve cell, then, as I said earlier, the sodium ions are pumped out of the nerve cells by an active transport. So active transport is the movement of ions or molecules against the concentration gradient. The concentration gradient with the use of ATP. And carrier proteins are involved in it. carrier proteins may also be known as pumps are involved in active transport. So let's see how carrier proteins or sodium potassium pumps are involved in an active transport. For example, if this is the phospholipid bilayer, this is phospholipid bilayer. Okay. So we have this phospholipid bilayer showing the polar heads outwards and the non-polar tails facing inwards. Okay. There are integral proteins that span the entire of the width of this phospholipid bilayer. So we can, draw this protein this way. Okay. So this is a carrier protein, maybe known as sodium, potassium pump, or a carrier protein. Okay. So this carrier protein has, I'll draw that here as well. You can see this is the same carrier protein, sodium potassium pump, but you see there is a difference in the shape at the different time. It has binding site here for 3 sodium ions. Okay. So 3 sodium ions can bind here on the inner side of this carrier protein. So this is a 3 sodium ions. Okay. Sodium concentration is low inside, low concentration, and sodium ion concentration is high on outside. But still, these 3 sodium ions bind with it. And also, there is an enzymes attached with this protein, which is known as, we can write that an ATPase enzyme. ATPase enzyme. An ATP molecule bind with it, which is broken down to, ATP is broken down to, ADP, an inorganic phosphate. This energy released is used to shape the change, to change the shape of this protein. So the shape changes from this shape, which has a binding site on inside, to this shape which has the binding site on and outside. और इसकी जो inner binding sites हैं, that you see that are removed, और वो change हो जाती हैं. So consequently, what happens? These sodium ions are moved out. So 3 sodium ions are moved out, and at the same time, 2 potassium ions bind here, and the shape, as the sodium ions are removed out of it and potassium ions bind with it, the shape changes again to this, and this process continue, and again ATP is used, and this would change the shape again. So this process continues for a while, removing the sodium ions out of the cell and taking the potassium ions into the cell against the concentration gradient. Okay, 2 potassium ions are taken into the cell even against the concentration gradient. Okay, so this is high concentration, of sodium ions on outside, and low concentration of potassium ions on outside, and low concentration and high concentration of potassium ions on inside. So sodium moves from low concentration to high concentration, and potassium moves from low concentration to high concentration into or out of a cell. This process is known as an active transport. Okay, so proteins which change their shape are known as carrier proteins, and this change in the shape is also known as conformational change or change in the conformation of proteins. Hope you have understood it. So let's see some questions on this part. Here we have this topic, active transport. We have a question here. Which statement defines an active transport? A movement of large molecules through the cell surface membrane into the cytoplasm. So you see there has not been any mention of movement from low concentration to high concentration. Neither there is any reference to use of energy. So this could not be a definition of an active transport. For an active transport, there should be the movement from low concentration to high concentration by use of energy. So movement of molecules or ions from where they are in low concentration to where they are in a high concentration. So this could be a correct choice, right? So ions movement or molecules movement from low concentration to high concentration is an active transport, but still, the use of an ATP has not been mentioned here. So let's see the other choices. Movement of molecules or ions from where they are in high concentration to where they are in low concentration. No, this is not possible. So this is not a correct statement. Net movement of water molecules across a partially permeable membrane from a region of higher water potential to a region of lower water potential. We have already done that. This is a definition of an osmosis, not that for an active transport. So then even if there is no mention of an ATP here, but still this statement is correct for an active transport. Okay. So B is a correct choice. We have another question. Antimycin is a chemical that inhibits the function of mitochondria. Which method of transport across the cell surface membrane would be inhibited by antimycin? So as antimycin inhibit the function of mitochondria, so the role of mitochondria is to provide ATP, which is used during an active transport. Facilitated diffusion doesn't need any ATP. Active transport needs an ATP, and even endocytosis needs an ATP. We'll discuss in the next class how ATP is needed for an endocytosis. So 1 and 3 is a correct choice. So C is a correct choice in this case. Now we have bacteria taken glutamic acid by an active transport. Describe the process of an active transport. This is for 3 marks. And it is the bacteria taken glutamic acid by an active transport. Describe the process of an active transport. It is a general question. There is no need to give reference of, the glutamic acid. Or even if you describe in terms of glutamic acid, that would be all right. So this is transport of materials or molecules, or you can say glutamic acid from their low concentration to their high concentration by the use of ATP. Okay. So as it is for 3 marks, you should look for the 3 points in it. You see, we have talked about the transport is against the concentration gradient. Like we discussed, from low concentration to high concentration, that means the same. Requires energy ATP. We have also described the use of ATP here. The use of membrane carrier transport proteins, we haven't mentioned this. And there is a conformational change, pump protein, or reference to specificity. We have not mentioned these 3 points. So if we write answer this way, transport of materials, molecules, glutamic acid from their low concentration to their high concentration, by the use of an ATP, we will have two points only. From low to high concentration, 1 mark, and by use of an ATP, 1 mark. But this question is for 3 marks. So we should refer through the carrier proteins. So that would be okay. This would be for 1 mark. Or if further uh, continue to write like the shape or conformation of carrier proteins changes by use of ATP. So this may be another point that you can write for this question. Now let's see. We have another question here. This also describes, this whole process of conformational change. As this is a cyclic changes in the conformation in proteins, we can start it from anywhere. So you see, we started from here. This is a phospholipid bilayer, which has, a carrier protein present here. This is the carrier protein embedded in phospholipid bilayer. And we have some molecules which have high concentration on outside and which have low concentration in the cytoplasm in a cell. Okay. So these molecules bind with it. These molecules bind with the carrier protein. And maybe this is for, this is calcium ion. So calcium ions bind with the binding site, which is a specific binding site for calcium on these carrier proteins. And it changes its shape. When the shape changes, the calcium ions are moved out of a cell. Okay. And to bring that back to their original shape, an ATP is used. Okay. So then an ATP binds with it and it is broken down to an ATP. So energy released from here is used to change the shape again. So this process continues and continues to as longer this protein is inactive, it continues to pump calcium ions out of a cell. So let's see what is a question on this topic. Cell surface membranes are involved with the movement of substances into and out of a cell. Calcium pumps in a cell surface membrane maintain a concentration of calcium ions inside the cytoplasm that is 1,000 times lower than outside the cell. Figure 1.1 shows the movement of calcium ions across the cell surface membrane. Figure 1.1 shows the movement of calcium ions across the cell surface membrane. So let's see a question. With reference to figure 1.1, explain why calcium ions do not pass through the phospholipid bilayer. That's very simple. We have already discussed it, that non-polar tails in the phospholipid bilayer provide hydrophobic barrier. Okay. So this hydrophobic barrier, which prevents hydrophobic barrier prevents calcium ions which are charged to pass through this barrier. Okay. So calcium ions are water soluble, charged. We have mentioned this point. And phospholipid bilayer has hydrophobic barrier. So these are 2 points that you should have mentioned in this case. Name and describe the process by which calcium ions are moved across the membrane. So in this case, it is an active transport, obviously. And why it is an active transport? Number one, carrier proteins are involved, which change the shape. Though the use of an ATP has not been mentioned in this diagram, but obviously we know that ATP is used, so we can give a reference to this change in shape is due to ATP used, and it moves calcium ions against the concentration gradient. Okay, so that's how you should have answered this point. Now, which of these processes allow movement in both directions across the cell surface membrane? Obviously, active transport can allow in both directions. Diffusion can allow in both directions, and facilitated diffusion in both directions, and osmosis in both directions, depending upon the concentration gradient and the need of the substances to take into the cell or to move them out of a cell. So that's all about an active transport. Now we have a bulk transport. As we discussed in the passive and an active transport, that movement of individual ions is not influenced by the movement of other molecules or ions in the medium. Ions or molecules will move down their concentration gradient or up their concentration gradient, and the carrier proteins involved are specific to even the channel proteins are specific to one or more particular specific ions or molecules. So they would allow only specific ions to move into or out of a cell. However, in bulk transport, it is not the case. The movement of individual molecules is influenced by the medium. So substances in a medium move in the same direction as that of the movement of the medium. Okay. There are two types of this bulk transport. One is an endocytosis, and other is an exocytosis. So let's see an endocytosis first. If this is, for example, an animal cell, maybe a phagocyte. So we will discuss how a phagocyte engulfs bacteria or engulfs foreign particle. If we represent this bacterium or this is a bacterium found in the body, the white blood cells gather or they move to the area where the bacteria are found, and these bacteria bind with the surface of a phagocyte. These phagocytes will have the complementary proteins to the antigens that are found on the bacteria. So they would bind. These may be known as ligand proteins. Okay. So there are ligand proteins on the cell surface membrane, which would bind with these bacteria. Okay. And then what happens? This is step number one. After this bacterium binds with cell surface membrane, the membrane folds inwards. Okay. So this membrane folds inwards. This is an inward folding of this membrane. This is number 2 point. Membrane folds inwards. And this captures this bacterium inside it. Okay. And then this membrane is separated. This is how this membrane is separated from this cell surface membrane and moves into the cytoplasm with this captured bacterium or a foreign particle in it. Okay. This membrane bound vesicle with the bacterium or with a foreign object trapped into it is known as an endocytic vesicle. And this way of taking materials into the cell by inward folding of membrane is known as an endocytosis. So this is an endocytosis. Intake of a material by inward folding of a cell membrane is known as an endocytosis. This material taken into the cell may be a liquid material or maybe a solid material, as it is the case in case of bacteria. So if this is a liquid material taken up by a cell, then it is known as pinocytosis. And if it is a solid material taken up by a cell, then it is known as a phagocytosis. So phagocytosis, that is for eating of a cell, and pinocytosis for a drinking of a cell. So accordingly, if this is a solid material taken up by by this cell in this endocytic vesicle, so this endocytic vesicle may be known as phagocytic vesicle or may be known as pinocytic vesicle. Okay, or may also be known as phagosome. Then what happens to this bacterium or any foreign particle that has been trapped by a bacterium into the cytoplasm? This is broken down by the enzymes which are released into, there are lysosomes present in this cell cytoplasm. So these lysosomes will fuse with this membrane-bound vesicle, which is called an endocytic vesicle, and the enzymes released by these lysosomes, which are known hydrolytic enzymes, will break down this bacterium into fragments. These digested products, like there may be proteins in the bacteria that are broken down to amino acids. There will be carbohydrates, polysaccharides in the walls or in a bacterium that will be broken down to the simple sugars. And you will also see some other materials like nucleic acid, which may be broken down into its basic components or the monomer units. Okay, then these digested products are absorbed into the cytoplasm, and still there may be some waste substances, undigested substances left in this vesicle, and these undigested or unabsorbed substances are expelled out of the cell. This vesicle fuses with the cell membrane. This is step number 1. And then from this vesicle fused with it, the contents are expelled out of it. And this process of expelling the contents out of the cell by fusing the membrane-bound vesicles on the inside of the membrane and then expelling the contents out of it is known as an exocytosis. This is an exocytosis. Exocytosis. Okay, this may be for the waste substances which are expelled out of a cell, or maybe for any secretory substances that are packed by or packaged by the Golgi bodies into these membrane-bound vesicles, and they are also expelled out. Maybe the enzymes, maybe the hormones, or maybe the neurotransmitters, or maybe other substances that are secreted out of a cell by an exocytosis. So this is an exocytosis. Now let's see some questions on these two topics. Here we have a question on an endocytosis. The diagram shows several processes taking place in a cell. Which processes are shown in the diagram and involve the cell surface membrane of the cell? Okay, so you see the contents are being expelled out here. So this is an exocytosis. Okay, and this is a foreign material being taken into the cell by inward folding of this cell membrane. So this is known as an endocytosis. Okay, so there is no movement through this phospholipid bilayer into or out of it, so it could not be, diffusion, osmosis, or an active transport. So this should be endocytosis and an exocytosis. So C is the correct choice. Then we have phagocytosis is the process by which bacteria are ingested by the cell. Describe the role of cell surface membrane during phagocytosis. So this is what we have already discussed. What happens? You see, the first step is bacteria antigen combine with or attach with the recognition by the receptor. So these are the receptor proteins or ligand proteins that are present on the cell surface membrane. Then antibody on bacteria combined with the receptor. This may be, we will discuss this point in immunity. Okay, there is no need to discuss it right now in detail. Similarly, these two more terms, opsonins, opsonization, etc., that we will also discuss in immunity. Reference to constant regions, or what is the constant region of antibodies or variable regions of antibodies. We'll discuss them in immunity in detail. So here now, what we need to discuss is this first point: bacteria attached to the receptors on the cell surface membrane, and then membrane folds inward or invaginates or envelops, engulfs, or encloses the bacteria or the foreign particle into it, and then membrane separates from here, accepts answer without membrane, where implies previously. So we have just mentioned, the membrane once, and then this membrane separates, this membrane bound vesicle separates as a vacuole or as a vesicle or as a phagosome or as maybe a phagocytic vesicle or endocytic vesicle. Okay. So simply, if we write an answer to this, bacteria or bacterium binds with receptors on cell surface membrane of phagocytes. Okay. The cell membrane then folds inwards to capture the bacterium. This folded membrane is separated as a vesicle from the cell surface membrane. So this is how you should have answered this question. We have phagocytic cells contain many lysosomes. Describe the function of lysosomes in destroying ingested bacteria. So we have discussed that lysosomes actually contain hydrolytic enzymes, and these enzymes may be proteases, maybe these are lipases, maybe these are carbohydrates, and they would break down proteins into amino acids, lipids into fatty acids and glycerols, and carbohydrates into simple sugars. Okay, by hydrolysis or by digestion. 2.1 is a scanning electron micrograph showing a macrophage engulfing some bacteria. Describe how macrophages engulf bacteria. That's the same question again. So only the image in the question that differs, but the rest of the question is the same, and we've answered that. Number one, it would bind with that. You see, there are many 6 steps given here. So the first point is that you mentioned that this process is phagocytosis and endocytosis. Then you mentioned that the bacterium binds to the cell surface membrane. Then you talk about infolding of the membrane, then separation, the fusion of the membrane to form vesicle or separation as a vesicle. formation of a vacuole, and this is an active process which uses an ATP. So that's how you can write more detailed answer. Explain the function of lysosome. That's the same we have already discussed. Now in exocytosis, which process would allow the movement of large protein molecule out of a cell? Okay, so out of a cell would obviously by an exocytosis. After final processing in Golgi bodies, collagen is released into the outer surface of cell by exocytosis. Complete the passage to describe the process of an exocytosis. After final processing the Golgi bodies in the Golgi body, collagen is enclosed in membrane-bound vesicles known as Golgi vesicles or secretory vesicles. And those membrane-bound vesicles move toward the cell surface membrane and fuse with the cell surface membrane on inside, and then expel the contents out. So that's how collagen is removed out of it. So see, here we have Golgi vesicles details, formation, packaging of the collagen in the Golgi vesicles. Then vesicles move, you can mention by the microtubules, or even if you don't mention through the microtubules, that's fine. They move towards the cell surface membrane and they fuse with the cell surface membrane, except plasma membrane too. And then alternative valid points budding from trans face of, we don't need to know the trans face of Golgi bodies or cis or trans face of the Golgi bodies. What are these details of fusion of phospholipid bilayer merge into one? You see this last point, if this is for example an animal cell, and here you see this is a Golgi vesicle which fuses with it, and then its contents are expelled out. So here you see how this fusion takes place. Actually, the phospholipid bilayers that are present in this membrane of the vesicle and the cell surface membrane, they fuse with each other. We'll discuss that in another question in detail. The table shows 3 processes that contribute to transport across cell surface membrane. Which processes are the result of random movement of the molecule? So random movement only takes place in diffusion and on osmosis, but not in an endocytosis. Okay, so that should be this C, the correct choice. Which statement about the components of cell surface membranes are correct? Diffusion can take place through lipids and protein pores. What is this? Which statement about components of cell surface membrane are correct? Diffusion can take place through lipids and protein pores. Through protein pores, it should be facilitated diffusion. I doubt. This is not a correct statement. Okay. The first part is correct. Through the lipid phospholipid bilayer, that may be fine. Endocytosis only involves lipids. No, not necessarily. Facilitated diffusion only involves proteins. Facilitated diffusion only involves proteins. Yeah, the facilitated diffusion only involves proteins. That should be correct. And osmosis only involves proteins. It doesn't involve proteins. So, and then this is, I don't think so. Yes, this is correct, 3. So this should be a correct choice. And this cannot be a correct choice. Why? Because 3 is not there. And then we have 2 more choices that have 3 in them. It means we will have to look for 4, because this is a 4 here and this is 4 here. So osmosis only involves proteins. No, osmosis do not involve proteins. Osmosis is through the spaces in the phospholipid bilayer. Okay. So if this is a phospholipid bilayer, osmosis is the movement of water through the spaces in between the tails of these, the phospholipids. So this is an osmosis. It doesn't involve any protein. So 4 is not correct in any case. So this is not correct. This is not correct. And this is not correct. So we have only this choice left with 1 and 3, but we will have to look for 1 again. So diffusion can take place through lipids and protein pores. Yes, it can through the protein pores, but then it would be known as facilitated diffusion. Okay, so through lipids, yes, this could be, like oxygen that diffuses through it. So this is diffusion. So that's a doubtful point. For diffusion through the protein pores, that would obviously be known as a facilitated diffusion. But as we don't have any other option other than C, so we will accept this statement number 1. So C is a correct choice. Hope you've understood it. So you might get sometimes such tricky questions where, the rejection method may be more valuable than finding the correct statement. You will have to reject the wrong statements actually. So this is wrong. We are confident, and endocytosis only involves lipids. So I'm not sure this. So 4 is actually, we are sure that 4 is not correct. So 4 is there, 4 is here, and 4 is in this option as well. So these 3 options are not possible. So that's very simple. That leaves only the C option to be chosen. Which processes that contribute to transport across cell surface membranes are active or passive? Endocytosis and exocytosis are active processes. Facilitated diffusion doesn't involve energy. Osmosis doesn't need an energy. So this should be a correct choice. A is a correct choice. Okay. So that's all about it for today. We'll continue with the cell signaling in the next class tomorrow. Thank you.